All right, welcome back. Today we're going to talk about Chapter 12, Genetics and Evolution, which is the final chapter in the general biology book by Kaplan. Congratulations, you made it to the end. It is also my least favorite chapter out of all of the written chapters for the MCAT. I hate this chapter. It is not fun. It's pretty boring. It's stuff that you've been talking about since you were 14 years old. So hopefully we'll tiptoe our way through it and we'll leave it behind us, right? Okay, so before we do that, for the people watching back home, make sure you check out below for the link to all of our recorded lectures and the link to the Drive folder that is associated with this lecture program, where you can find my study guide, a sign-up sheet for our email list, as well as all of my notes. And tonight, I'm going to be uploading my chemistry notes, because after this, we will be starting General Chemistry, one of my favorite books that Kaplan has ever written. Not because I'm biased or anything, but chemistry is the greatest. All right, so that being said, I have uploaded, I think, up to Chapter 9. I have Chapter 10 and 11 ready for upload on my computer. I'll probably get to that either tonight or tomorrow morning. And then this will be uploaded as quickly as possible because there was a slight schedule change at Brooklyn College, but hopefully you guys will be able to watch back home. That being said, let's begin. So in order to begin our discussion about genetics and evolution, we are first going to talk about genetics, right? And the fundamentals of genetics begin with genes. Right? And genes, these are DNA sequences that code for heritable traits. Right? DNA sequences that code for heritable traits. So, inside of my DNA somewhere, there's a trait, a gene, right? A sequence of DNA that codes for my dark brown eyes, and my black hair, and my height, and the way that the hair grows on my arm, right? All of these things are heritable traits, right? And they were passed on to you from the people who produced you, right? Your biological parents. All genes are organized into chromosomes, right? And since we have pairs of chromosomes, we have one specific sequence for each gene. Does anyone know what that little one sequence, that one unique sequence is called? It's called an allele, right? And then each chromosome carries one allele for a gene. Well, what's an allele, right? What's an allele? They're just forms of the genes. So basically, inside of me specifically, right, I have an X chromosome, right? And then I also have a Y chromosome. And on that X chromosome, there is a gene that codes for a specific type of cell inside my eye. But that gene is defective, right? Which makes me colorblind. So the gene is, well, that gene is a specific allele that codes for colorblindness. And since I don't have another one, that's the one I display. But in my sisters, right, they might have one good one and one bad one. So they have a good allele and a bad allele, a defective allele and a normal allele, or what we call a wild type allele, a non-mutated, right? Okay. Everyone gets like the whole thing about alleles and stuff like that, right? And then we just have some short discussion, genotype and phenotype, right? So genotype is the way that your actual genes are assorted, and the phenotype is the manifestation of those traits. So I have a certain DNA sequence that looks like blah, and another DNA sequence that looks like blah. And over here, this is my genotype. And those two together give me black hair. And black hair is the phenotype, right? Cool. What else? A homolog is one of two of the same chromosomes passed on by a person. We read that in homologous chromosomes, right? And then a locus is a gene's position on a chromosome, right? So if you have a chromosome that looks like this, and you have a gene right here, that position can be described, right? And that's that gene's locus. That's where the gene lives. It's like its address, right? Cool. All right, let's talk about dominant and recessive alleles, right? So, dominant alleles. What's special about them? If owned, then expressed. If you own the allele, you express it. Right? If you own the allele, you express it. I have to look up something for an example, because it's a very good example. Okay. Apparently it's a myth. I was about to talk about the widow's peak thing, right? Because I have widow's peak and some people don't, right? What is widow's peak? It's the little triangle that at the bottom of your hairline. I have widow's peak. Some people don't like it, right? I don't really care for it. But it's said that widow's peak is dominant and then not having widow's peak is recessive, right? But apparently that's a myth that I just looked up. But it's like I can't really think of other dominant and expressive things, but we'll get there when we get there, right? Okay. So a dominant allele basically means that, let's say that, and this is an example, let's say that there was a tadpole, right? And that tadpole could either have a white spot on its back or not have the white spot on its back, right? And let's display that with the letter W. Actually, let's display it with W. So a big W and a little w, right? The big W is usually used to display, or capital letters usually used to display a dominant allele. And then the lowercase is for the recessive allele. Let's say that having the white spot was dominant and not having the white spot was recessive. Right? So if a tadpole was born that looked like this, it would have the white spot. If a tadpole was born that looked like this, it would have the white spot. But if they were born like this, they would not. And that brings us to the idea of recessive alleles. Only expressed if both alleles are recessive. Going back on the point of someone who's female with color blindness. For me, it's not that tough because I've only got one place to fuck up, right? But for a woman, you got to fuck up both of them to give them color blindness. So that is like what we call a recessive trait. And this is also called what? Does anyone know? When two of the same genes are expressed the same way in the genotype, homozygous, right? Homozygous. So just for vocab, this is homozygous dominant. This is homozygous recessive. And this is heterozygous. Right? Because homo means one, or one type, and hetero means both, or two types. There's also something known as hemizygous, and hemizygous means there's only one allele. It's rarely ever brought up. Okay. Now, let's talk a little more about dominant alleles, because recessive alleles are easy enough, right? But dominant alleles, they can get a little tricky. Because when something's dominant over something else, it's more powerful than that thing. But what happens when more than one dominant things come together, right? So this is more like complete dominance. I was talking to one of my engineering friends while I was studying for this. I remember I was on FaceTime with him and I was studying like complete dominance, blah, blah, blah. He's like, so is the other allele called submissive? I was like, no, it's not called submissive. All right. So this is the first special clause. More than one allele is dominant and both are expressed. What's that called? Codominance. What's an example? AB blood. A antigen, B antigen, same red blood cell. No antibodies. What was the big whoop about AB blood again? They are the universal receivers. Receivers, right? Specifically AB positive people, right? Right? And then, if a heterozygote, well, this isn't really dominance, but let's say another special clause over here. A heterozygote leads to expression of both alleles. That's called incomplete dominance. For example, pink roses. Pink roses. Right? Because capital R dominant is red, and lowercase r recessive is white. So you mix the two, you get incomplete dominance, and you get pink flowers. That make sense? Yeah? You all snap a photo? All right. Anyone have any questions about dominant and recessive alleles? Cool. Stuff we've been talking about for a couple years now, right? No. They go into a little tangent about penetrance and expressivity. I'm not gonna lie. I'm not the best at describing what penetrance is, but we'll give it a shot. Penetrance. By textbook definition, it is the proportion of individuals in the population carrying the allele who actively express the phenotype, right? So it's the proportion of allele carriers who actively express the phenotype. So, how many people have the allele and how many people express it? And normally it's express over own, right? It's a proportion. So this is where it gets kind of interesting. So alleles are basically classified by their penetrance, right? And they give you this whack example, and I'm just going to dictate you through it, because this is basically all you need to know. There's a disease called Huntington's. You guys have heard of it, right? It's a nucleotide repeat disorder. So what happens is that inside of your DNA, you have these little sequences, right? Let's say that it's like A, G, C, T, U, T, right? Like a sequence like that, right? Or, stupid. Sequence like that inside your DNA. This is a repeat sequence. And in some people with Huntington's, example, I don't know the actual nucleotide repeat. I will have to know when I take my STEP exam, but I don't know it now. this thing repeats like 40 times inside of their DNA. Now, in respect to how much DNA you have, that's not that much, right? But when people have 40 times of that repeat, we say that the allele has 100% penetrance. What does that mean? That the people with the allele for Huntington's, with that number of repeats, have 100% penetrance, which means all of them express it, right? But it's possible that people with like 30 repeats instead, some of them are asymptomatic and some of them do show Huntington's. So then the penetrance is like 65% or something like that. Does that make sense? You guys get where I'm coming from? So alleles can have severity, or they can have variable expression in the number of people who show that allele. And it doesn't only have to be for bad alleles, it can be for neutral alleles as well, right? So the penetrance is basically saying how much is something that's inherited actually displayed to the rest of the world. That's what we're saying. I know it's not the best explanation, but I'm not a geneticist. So if this proportion is 100, it's called full penetrance, right? And then a person with fewer repeats than 40 would have high penetrance, and then a person with fewer repeats than that would have low penetrance. All right, expressivity is a little easier. We have two types of expressivity: constant and variable. Constant means all individuals with a given genotype have the same phenotype. A plant that is AA and another plant that is AA look the same for that gene. They display that gene the same way. Variable expressivity, individuals of the same genotype may differ in phenotype. May differ in phenotype. Yeah? Let's talk about some old white dudes. I'm not kidding. Let's talk about Mendelian concepts. Who is Mendel? He's like the father of genetics, right? So there's a first law and there's a second law. The first law is called the law of segregation. There's a couple parts to it. Genes exist in alleles. An organism has two alleles for each gene. The two alleles segregate during meiosis. And heterozygotes display the dominant allele. Second law. Independent assortment. inheritance of one gene does not affect another. Is that true? Always? No, it's not always true. So that's what we call Mendelian genetics. When things follow these rules, they express Mendelian inheritance. I was texting Professor Malitsky the other day, and I told him about this whole program and everything. And the one thing I asked him, I was just like, how do I make my handwriting better on the board? And he's just like, well, you might want to teach for like 15, 20 years, and then you'll get used to it. Well, hopefully in 15, 20 years, I will not still be here. And here's hoping I will never have to see any of you in 15, 20 years, because that means you're very, very, very ill. I love it when people are just like, they talk to medical students, something they say all the time. It's sweet. They're just like, oh, like I hope I see you when you're a doctor. And I'm just like, I don't. You know, see me outside of work? That's fine. I was talking to my buddy David the other day. He lives up in Buffalo with his girlfriend. And he's just like, yeah, man, like when you're a surgeon, like free surgery. And I was like, fuck no, dude. I don't trust myself to do that. If I fuck up, that's on me. Then I lose my best friend. All right. They want you to know a little bit about like the origins of DNA and experimentation. I don't think it's asked about all that often. Once we get to biochem, we're going to talk about a little bit of the background of how all these experiments work. But do you guys remember like Frederick Griffith, Avery McCarty and McLeod, and then Hershey and Chase and all their experiments and everything? It comes back up in biochem when we talk about the actual synthesis and sort of passing on of DNA and stuff like that. So we're going to glaze over it now and just look at it later. But Frederick Griffith, it was the transforming principle. So it's like if a virus was injected into a mouse and then that was passed on, like the virus gets passed on with DNA, blah, blah, blah. Avery, McCarty, and McLeod, they analyzed bacteria treated with enzymes to break down the DNA, and then they found out that DNA was the transforming particle for like bacteria and stuff like that. And then Hershey and Chase, that was the cool one. So it was radioactive sulfur in proteins and then phosphorus in DNA, and then they traced it. So they were just like, okay, well, sulfur only exists inside of proteins, and then phosphorus pretty much exclusively exists in DNA. Does anyone know why sulfur is in proteins? What is inside of proteins that has sulfur in it? Take a crack at it. Yeah, disulfide bonds, exactly correct. Well, there's enough of an answer, right? But the disulfide bonds specifically, they come from an amino acid known as cysteine. That's cysteine, right? And there's another one called methionine, and methionine also has a sulfur group on it. But this one's the important one for disulfide bonds, because what happens is it hooks up with another cysteine right here, and then you can get a disulfide bond. Anyone know where disulfide bonds are very prevalent in your body? Your hair. Your hair. I made a whole TikTok about how a perm works. And basically a perm is when you break those disulfide bonds, fix your hair into a certain position, and then reform them. And it reforms all the disulfide bonds in those curls, and that's how a perm works. Short tangent. Changes in a gene pool. What's the biggest one? It's called a mutation. Right? It changes an allele to something different than the wild type. Things that cause mutations are called mutagens, right? And one more thing, there are things called transposons. And they're basically fragments of DNA that can be inserted somewhere else. If that falls into the coding region of another gene, it can cause a mutation. If you're following along with my notes, please take a look at this diagram right here. Or if someone next to you has it open, look on theirs. So there is a normal strand of DNA, and we are talking about nucleotide level mutations, right? There is a normal strand of DNA. And what happens is that at some point there is a codon. What's a codon? It's 3 base pairs, right, inside the DNA. Or 3 bases. And the codon is TTA. Thymine, thymine, adenine, right? And TTA, when it's transcribed and translated, it codes for leucine. What is leucine? It's an amino acid, right? This poor fuck spent too much time inside of a tanning salon, right? And their TTA was changed to TTG, right? But that still codes for leucine. This is called a silent mutation. And silent mutations normally happen on the 3rd base of the codon. Why? Because if you look at my notes, I wrote a word right there that says wobble. And that's what it's called. It's called wobble, right? That a lot of the codons, if you look up on Google, if you look up a codon chart, right, a codon translating chart, a lot of the codons will have this same first 2 letters, and then the 3rd letter doesn't really make a difference. Why? It's to protect against shit like this. That's why we have wobble. All right. Good? That's a silent mutation. What's a silent mutation? It's a mutation without a change in final protein structure. When I'm saying proteins, what do proteins have to do with this? We're talking about DNA. Well, DNA codes for RNA, and RNA codes for proteins, right? So it's a mutation in the DNA without a change in the final protein structure, right? Now, things can fuck up along the way, right? Like the mRNA can get degraded, or the protein might be misfolded, or like the amino acid might not deploy properly, blah, blah, blah. Those are not called mutations. Those are errors in protein synthesis. These are called mutations. So that's a silent mutation. The next one is when TTA becomes TGA, where TCA, where TTA made leucine, TCA makes serine. They're different. They're not the same. They're not the same, right? And that means this is a missense mutation. So it's when a mutation changes the amino acid coded for by that codon. How many amino acids are you responsible to know? 20. You're responsible to know 20 amino acids. And we'll go over all of them when we go over biochem. But if you're studying by yourself and you want to know how I memorize my amino acids, every single time I encountered a word, like let's say glycine. I had to write it down, glycine. Next to it, I would write down GLYG, the 3-letter code and the 1-letter code, every single time I write it down, right? Every single time. Because most times, you won't see this. You'll either see this or you'll see this, and you'll have to recognize that that's glycine, right? And some of them are easy like that. Some of them are not, right? Because aspartic acid is D. It's not A, because A is alanine. It's already taken, right? Cool. And then what else? You can go back and just look at the amino acid and be like, do I know how to draw a glycine? Well, hopefully, because it's the only one with nothing on it, right? And sometimes you just build up from glycine, alanine, phenylalanine, tyrosine, right? Just keep building. That's my little note on amino acids. Anyways, missense mutation. Go from leucine to serine. It's a pretty bad mutation. Why? Because leucine is neutral, and then serine has an OH group. So now you've fucked up the chemistry of the protein you're trying to make. All right. Next. This one's, these next two are interesting. So TTA turns into TGA. And this is leucine. And TGA, when you translate it, it turns into UCT or something like that, whatever the fuck it turns into. I don't know if this is the coding or the non-coding strand, but this translates to a stop codon. So in the middle of what's supposed to be the protein, it stops the synthesis. It stops the synthesis, right? And this is the coding strand. It just says it right there. That is called a nonsense mutation. What happened there? You are trying to build a big old skyscraper, right? And then all the people you hired to work for you all get murdered on the same day. You just kill all of them. Now your building's only this tall, when it's supposed to be that tall. That's nonsense, right? The fuck happened? So the protein is supposed to be like 300 amino acids long. We stopped it at 6. Tough shit, right? It's a premature stop codon. The last one, I'm not going to be able to draw it out. It's called a frame shift mutation, right? Actually, I can write it out. I'll show you what I mean. So let's say like ACTGATGCA, right? And we go triplet, 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 right? And we take this A right here, and it gets deleted. So what happens? ACTGTGCA. Whereas ACT and GAT would code for specific, so A1 and A2, right? Amino acid 1 and amino acid 2, or let me write it out properly. Now we have AA1 and AA3. Because GTG is not the same as GAT. And now we don't even have the rest of the code over here. It's all going to get shifted. So whatever happens after this, GCTATCG, right? Now instead of GCA, it's CAG. So do you see how the whole thing gets fucked up by deleting one base pair? That is called a frame shift mutation. And they are devastating. Because it could make premature stops, and it could make misfolded proteins and random strings of amino acids that aren't even supposed to be in the body. That's a problem. Okay? Now, silent mutations, missense mutations, and nonsense mutations are also called point mutations. Because it's that one specific base that gets changed. Why is this not a point mutation? Because it's a base that gets deleted, right? All right, cool. So those are DNA mutations, specifically at the level of the DNA. Let's talk about some chromosomal mutations. Some of these are self-explanatory, like a deletion. You guys read back there? Yeah? A deletion is when a segment of DNA is deleted. Or a duplication, where a segment of DNA is duplicated. Or an inversion, where a segment of DNA is reversed. You guys get the point? Right? All right, now those are the three that sound the same, right? Now let's talk about the special ones. Yeah, you guys don't need this? Yeah, it's just deletion, duplication, inversion. What happens to the DNA segment, right? Now let's talk about the two special ones. Because, like Professor Malitsky always said, there's always a special case. An insertion. So a segment of DNA is moved from one chromosome to another. And this is why they're special, because they sound so similar, but they're completely different. There's also a translocation. Two chromosomes swap DNA. All right, ready? I'm going to show you the difference. This is chromosome 4. And this is chromosome 5. Okay? An insertion is this. That's an insertion. That bottom part moved from chromosome 4 to chromosome 5. A translocation is different. This is chromosome 15, this is chromosome 16. After the translocation, this is chromosome 15, and this is chromosome 16. They swapped parts of themselves. That is different, and entirely different than crossing over. Don't get that mixed up. Crossing over is when you take the same genes, the same alleles, the same parts of homologous chromosomes and swap them with one another. This is chromosome 16's tail on chromosome 15's head. That's like if I chopped your legs off and chopped my legs off and swapped them. It's pretty fucked up, isn't it? Call me fucking Frankenstein. Translocations are not good. They're causes of very, very, very serious, debilitating congenital illnesses. And if you're religious, or whatever you believe in, just thank God that it's not you. Some mutations are advantageous. Some mutations are not so advantageous or deleterious. See you next time. There's a special class of mutations that the MCAT loves to talk about, and are very, very, very important in medical school. They are called the inborn errors of metabolism. You will read a lot of passages about them. And one of them is phenylketonuria. It is a mutation that reduces the activity of an enzyme called phenylalanine hydroxylase. You'll read about this a lot. The MCAT loves to refer to this specific disease. And since phenylalanine hydroxylase has a decreased activity, you get a buildup of phenylalanine and then a buildup of byproducts of phenylalanine called phenylketones. And those are toxic. Phenylketonuria. Phenyl, like benzene, phenyl. Ketone, as in ketone without the E. And urea, meaning in the urine. Phenylketones in the urine. That is a classic hallmark of the disease. Yeah, what's up? It's an enzyme. Phenylalanine hydroxylase. Yeah. So what it does is it hydroxylates phenylalanine into tyrosine, right? And the buildup of phenylacetate and phenylactate inside of the bodies and bloodstreams of people with this disease cause many, many, many disastrous effects, including growth stunting, mental retardation, bad body odor, motor problems, etc., etc. It's a very bad disease. The MCAT just brought it up. I read like 3-4 passages just about this. And what they want you to know is that there's a mutation in a chromosome that is passed on, right? And where did the mutation likely occur? Probably happened in a sperm or an egg cell, right? It's passed on, and you're born with it, and then you get the fucked up enzyme, and now you're just screwed. It's called an inborn error of metabolism. And the reason we make the distinction is because once you're born, even when you're a couple of days, couple of weeks, couple of months old, that gene manifests, right? But there are some mutations that take years to manifest. Those are not necessarily called inborn errors of metabolism. That's a metabolic thing, where when your metabolism starts to kick off after you're born and after your body starts working, you're just fucked. There's a bunch of them. Glycogen storage diseases, lysosomal storage diseases, blah, blah, blah. A lot of these you might have heard of because a lot of them are very prevalent in the Ashkenazi Jewish population. Lysosomal storage diseases have very high prevalence in that specific population. They are genetically predisposed to those sorts of conditions. African Americans predisposed to hypertension. South Asians predisposed to high cholesterol. Eastern Europeans predisposed to certain types of skin cancer and Alzheimer's, right? These are all genetic predispositions of different ethnic groups that exist within our society, right? Okay. Let's talk about genetic drift. Genetic drift. So they are changes in the gene pool of a population due to chance. For example, the Fowler effect. Isolation yields evolution independently. Let's say that you have a population of people living on an island. All these people. They look so happy, don't they? And each person I drew here stands for 100,000 people. So there's 400,000 people living on this island, right? The island, unfortunately, splits in half. And they go their separate ways. Now, that population is going to evolve differently than that one. That's called the founder effect. Isolation leads to evolution independently of one another. Okay. One thing they talk about is inbreeding, which yields increased homozygosity. Why? Well, let's look at a couple generations of people inside of a Punnett square. You guys all know what Punnett squares are, right? I don't have to go over that or anything. Cool. So let's say that there was only one person with a recessive allele inside of a population, right? You know, these two people got together, had kids. Right? No one's expressing anything yet, right? Let's say just by chance, obviously hopefully not the two people who had the same children, but whatever. Someone else who looks like this, someone else who looks like this, got together, had kids. It'd be the same thing, right? But eventually, after a while, by chance, there would be a mixing of two people like this. Uh-oh. And that person would end up mixing with this person. Even bigger uh-oh, now everyone's either carrying or they're expressing. Now this doesn't necessarily have to be a bad thing, but in a lot of cases it is. Right? And that's why inbreeding and homozygosity is said to lead to decreased population fitness. Like the Habsburgs. You guys know who the Habsburgs are? If you don't, look it up. It's pretty interesting. So outbreeding would be a good thing. Because when you outbreed, you're not communicating those same genes and those deleterious things that are within your population. So let's say that I have a kid someday. And I'm South Asian, and I marry a South Asian woman, and we have kids. And then our kids carry those genes that code for high cholesterol and hypertension and stroke and blah, and whatever the fuck else South Asian people have. But then that kid goes and has children with someone of Western European descent, who has the whole recessive genes for Alzheimer's and this and that and blah, blah, blah. Since my kid has the recessive genes X, Y, Z, and then that other kid has recessive genes A, B, C, isn't there less of a chance that they're going to end up making a kid with those diseases? Not just that, but there's also more of a chance that these genes will disappear over time. Because of what? Like I said before, right? But if this now these two have no chance of passing it on. And these two have like a 25% chance of passing, depending on who they mate with, right? So do you guys see how like outbreeding can kind of decrease that percent chance of giving on a bad gene? This is why outbreeding is said to increase population fitness over time. Just by chance. It's just probability. I am not teaching Punnett squares. Learn that shit by yourself. Because eventually, like, the Punnett square, it's fun, it's easy, blah, 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 and then you get to the dihybrid cross. I'm not doing it. It's not happening. All I remember is, like, the 9-3 to 3-1 or some shit like that. I don't even know what that means anymore. God. All right. So, one more thing that comes up every now and again is gene mapping. So genes that are close together are more likely to be inherited together, and genes that are further apart are more likely to be inherited independently. Independent assortment. This is non-Mendelian genetics, right? So the likelihood that two alleles are separated from one another during crossing over is the recombinant frequency, and it's proportional to the distance between the genes on the chromosome. So what was crossing over again? It was when this guy, chromosome 4, and this guy, chromosome 4, swapped their chromosome 4 things to both be chromosome 4, right? So they swap heads and they swap tails, and they're both still chromosome 4, right? But remember, that copy of chromosome 4 came from the mom, and that one came from the dad, and then that tail came from the mom and the dad, blah, 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 crossing over, whatever, right? Now, if this portion of chromosome 4 is swapped with this portion of chromosome 4, and there's a gene A that exists here and a gene B that exists here, they're going to get inherited together by whatever fucking fetus inherits that gene, right? But if there's a gene C over here, and that's the part that's getting crossed over, gene C was just now detached from gene A and B. Why does that matter? Because it's not A and B over here, it's A prime and B prime and C prime. So now when they swap over, it's going to be C and then AB prime, and it's going to be C prime and AB from the two different people that the crossing over happened from. So the distance apart of C to A or C to B, the greater it is, the higher the recombination frequency. And we measure that recombination frequency in a variable called theta. Do you guys want to run through Hardy-Weinberg? Does that matter? No? It's pretty straightforward. Basically, it's like P plus Q equals 1, and then P squared plus 2PQ plus Q squared equals 1, right? And basically what this is saying is these are the homozygous dominant, the heterozygous, and the homozygous recessive. And then this is the frequency of the dominant allele, and this is the frequency of the recessive allele. They're going to give you numbers, you plug them into the equations, you solve for whatever's missing. That's it. Okay, that's as easy as it'll get for Hardy-Weinberg. If you don't understand it now, when we do practice problems, they'll come up, we'll chat about it. It's literally just a little bit of algebra. That's it. But remember, this is used to measure allele frequency, and the MCAT does ask about this every now and again. Given that mating is random, population size is large, there are no mutations, no migrations, and all genes are equally successful at being inherited. So basically, if they follow Mendelian genetics. That make sense? Okay. Just a couple terms to talk about before we wrap up the chapter. It's a quick one. Then we're going to start chemistry. Can you guys tell I'm excited to move on, to get done with this? Natural selection, you guys know what that is? Organisms that have better mutations and better fitness will survive and mate better, and then organisms with less mutations that are beneficial will not survive and be killed out of the gene pool. Does that still happen? Of course it does. That's why the human population, we're constantly making better athletes, better runners, better this, better that. It's also medicine and science and this and nutrition, blah, 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 right? It does happen. Modern theories like neo-Darwinism adds the knowledge of genetic inheritance to Darwin's original theory. So when a mutation or recombination results in a change that is, I can't even read what I wrote, favorable to an organism's reproductive success, there's a chance that that's passed on to the next generation. So basically, it's the same thing, right? You have a mutation, it gives you some benefit, there's a higher chance that mutation's passed on. It's just like the recessive versus dominant thing we were talking about before. If you have it, you mate with someone, more chance that you pass it on to your kid, more chance your kid passes on to their kid, blah, 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 blah. Right? Modes of natural selection. Okay, these are actually kind of important. This is a chart. And I hate this chart because it's not labeled in the fucking textbook. That's the number of individuals, but I'm assuming this is a phenotypic display, all the phenotypes that can be expressed, right? So one of them is called stabilization. And it's when the population kind of stabilizes towards this middle point. So what is this? An example here would be high and low fetal birth weight are selected against. Fetuses with low birth weight die. Fetuses with high birth weight kill their mothers. So fetal birth weight kind of standardizes towards the middle. Right? One of them is called directional selection. Antibiotic resistance in bacteria. All of them want to go that way. Want to go away from not being resistant. All of them want to be resistant. And the last one is called disruptive selection. This one's cool. So this is beak size, narrow and broad, right? So finches like narrow beaks and broad beaks, but not really things that are in between. Because narrow beaks are easier to catch worms, and broad beaks are easier to break acorns. So it makes it easier to eat. Over here, this would be less resistance, more resistance. Over here, this would be lower weight, higher weight. So it's all the difference in the phenotype. Yeah, what's up? Narrow beaks and broad beaks in finches. What are finches? They're birds. They're the birds that Darwin was looking at. So in finches, broad beaks for seeds and narrow beaks for worms. But being in the middle doesn't really help you, because there are no worms that are as hard as seeds, and there are no seeds that are as narrow as worms. It's also a small term called speciation. It's just the accruement of different mutations over time that produces a different species. Last thing, and then we're finally doing chemistry. So fucking hype. God, I can't wait. I know, right? So exciting. All right. Divergent evolution, parallel evolution, and convergent evolution. This is independent formation of dissimilar characteristics from a common stem. This is related but evolve independently but similarly in response to environment. And this one is independent formation of similar characteristics. They don't really give you examples for these, unfortunately. What time is it? 7:03. Really? Oh, thank God. This is divergent evolution, so it's independent formation of dissimilar characteristics from a common ancestor, from a common stem. Well, guys, you'll be happy to know that that is all there is to know about biology, for the MCAT at least. So congratulations, you guys made it. You can clap for yourselves, but not for me, because my job's not over. All right, so that is chapter 12. We'll get this uploaded as quickly as possible. I have my notes up on biology done. We're going to fill out the spreadsheet. Make sure you check the spreadsheet for all the links and recorded lectures. Now, in the spreadsheet, you guys know how Google Sheets work, right? We're going to have a sheet for biology. There will be another sheet for chemistry, and then another sheet for thing, all in the same sheet file. They're just on different pages. All right? Cool? Thank you guys so much for keeping up. Really proud of you. Wouldn't be a class without all of you. Let's do some chemistry.